morning, viewer. Welcome to our Globe viewers of breaking news. A new study from the MRC Laboratory of Medical Sciences in London, UK reveals how ancient viral DNA, once written off as junk, plays a crucial role in the earliest moments of life. The research published in Science Advances begins to untangle the role of an ancient viral DNA element called MERVL in mouse embryonic development and provides new insights into human muscles wasting disease. Transposable elements are stages of DNA that can move around the genome. So many of these DNA sequences originate from long ago when viruses inserted their genetic material into our ancestors' genomes during infection. Today, today these viral transposable elements make up around 8 to 10% of the mammalian genome. Once disregarded as junk DNA, we now know that many transposable elements play an important role in influencing how genes are turned on and off, especially during early development. So they have a variety of beneficial and harmful roles in the body. For example, some help regulate normal immune responses while others can disrupt genes. The latest research led by Dr. Mechalae Parksor of the Chromatin and Development Group at the LMS, or former postdoctoral researcher Dr. Paul Chamas, focuses on a viral transposable element called MERVL. So this element becomes highly active for a short window of time when a mouse embryo reaches the two-cell stage, the point at which a fertilized egg has divided into two cells and switches on its own genome for the first time. Cells. Cells in this state are considered totipotent, meaning they can generate every cell type of the embryo and extra embryonic tissues like the placenta. Viewers, MERVL acts as a central switch to activate a large network of genes specific to the two-cell stage of development, but its precise contribution is not well understood. To work out the role of MERVL, the team used a gene manipulations technique called CRISPR activations to turn on MERVL elements in mouse embryonic stem cell to mimic what happens in two-cell embryos. In cells where only MERVL was activated, the cells looked like they were only partially similar to cells of the two-cell stage, but they still had several characteristics of totipotency. So the researchers described this in-between state as an intermediate phenotype. They showed that activating MERVL alone is sufficient to create totipotent features in early embryonic development. MERVL is activated by a transcription factor, a protein that helps genes turn on and off, called DUX. While DUX is important for switching on the two-cell developmental program, when it is active for too long, it becomes toxic to cells and causes cell death. We have the exclusive report.